Hello, very happy good evening. So this is Krishnaveni and we've been talking about the story of life on earth, and that's biology. So welcome to my class today, that is Human Reproduction Lecture 3. So today we are going to discuss about the process of fertilization and implantation. I'm really sorry for being extremely late. So there was a technical issue and it took us some time to get out of it and resolve it. So I'm really sorry for the inconvenience. I'm abundantly grateful for all of you for waiting for me for the past 20 minutes. So that really shows how dedicated you are. So thank you so much, and this made my day. So you guys were asking me, so you guys were giving me the patience to stay calm because yes, it will get resolved. So your messages made me wait patiently with them, and yes, finally it got resolved. So thank you for patiently waiting with me. So thank you once again from the bottom of my heart. So today we're going to talk about fertilization and implantation. So hi Anthony, a very happy good evening. Hello Nausheen, so welcome to my lecture. So this is your first lecture here, so please don't forget to like, share and subscribe. So hi Dharini, yes, a very happy good evening. Hello Naveen, so Naveen thought he is late but ma'am was actually late. So yes, thank you so much for giving this cooperation to me. So hi Vinay, yes Shubham sir, that's fine. Okay, so here we go into human reproduction. So this is lecture 3. So we were talking about the structure of the male and the female reproductive system, and then we went forward to talk about gametogenesis. What is spermatogenesis? What is oogenesis? What is your menstrual cycle? Now we're going to talk about the process that happens further, that is fertilization and implantation. Yes, a very happy good evening, and thank you everyone for joining my lecture. So please don't forget to like, share and subscribe. So shall we begin? Yes. Before we begin our lecture today, so tomorrow at 5 p.m. we are completing the last chapter in human physiology, that is chemical coordination and integration. So after the completion of human physiology chapter, so we will have a mentee only on human physiology. So make sure you prepare well for that mentee because that will give you a lot of confidence because human physiology has a high weightage, right? So we will have the mentee probably on Friday or Saturday. I will let you know tentative dates. Okay. Fine. So human reproduction. So here we begin with fertilization and implantation, lecture 3. Okay, so you have utilized this weekend very productively. So that's absolutely nice to hear. So you have revised well. That's very good, Anthony. So you are having a very positive mindset. You're staying healthy. Very good. So please spread positivity. So that will give you a lot of good approach. Yes, thank you. So which means every Saturday you need a motivation. Is that so? Yes, definitely. So from Thursday we are beginning plant physiology. So I will start with photosynthesis, respiration. Then I'll go to the other chapters. Okay, Navi? Yes, your suggestion is taken. No issues. Fine. So human reproduction. So what are the topics that we were talking? So we were discussing about the 7 steps in human reproduction. So we were talking about your male reproductive system. Then we went on to talk about your female reproductive system. Right? So then we were talking about gametogenesis. So gametogenesis includes 2 things. So this includes your spermatogenesis and your oogenesis. Right? So menstrual cycle we spoke about. Then we are about to talk about fertilization and implantation. So then we will talk about pregnancy and embryonic development, parturition and lactation. Hello Shimpi, a very happy good evening. So thank you so much for joining my session. Yes, I'm absolutely fine. How are you? I heard you couldn't come because of some issues at home. I hope everything is fine. All of you are fine. Yes? So thank you so much for joining my session today. So please do like, share and subscribe. So let's see if we can complete this chapter today. If not, I will take an extra class and finish it. That's not an issue. Okay. So here we move forward. So I will revise from gametogenesis. So we are supposed to have a recap of gametogenesis. So gametogenesis is the process of formation of gametes, right? It is the process of formation of gametes. So the gamete in your male is your sperm and the gamete in your female is your ova. So how does the process happen? So in male we call the process as spermatogenesis. So the process of formation of sperms, right? So this spermatogenesis process begins at the time of puberty. So puberty is the stage of sexual maturity, right? So what happens in spermatogenesis? So here the primary sex organ in your male, that is testis, comes into picture. So testis has nearly 250 compartments, which is known as testicular lobules. So in each of these lobules, you have 1 to 3 highly coiled structure. So these structures are known as your seminiferous tubules. Your seminiferous tubules, if you remember, has two structures. That is your male germ cell, which is also known as spermatogonia, and your Sertoli cells. The Sertoli cells nourishes the growing sperms, but your spermatogonia here is going to play a role. So your spermatogonia is plural, the spermatogonium is singular. So this undergoes mitotic division to increase its number, so that half of the spermatogonia becomes your stem cell. Okay. So the remaining half is ready to undergo meiotic division. So the one that is ready to undergo meiotic division, okay, so this is your primary spermatocyte. So primary spermatocyte is diploid. So this undergoes meiotic division to form two haploid secondary spermatocytes. So this is haploid, this is haploid. Now it undergoes meiosis division too, and it forms your spermatids. So all these are haploid. So spermatids becomes your sperms or your spermatozoa. So here this process is known as Spermiogenesis. Okay? Yes, Anthony, definitely I will get back after class. Okay? Yes. So now it is released out from the seminiferous tubules. So here this process is known as spermiation. So this is the process of your spermatogenesis. So here what is important is you should remember the ploidy. So one spermatocyte can give you 4 spermatids. So spermatids are haploid, sperms are also haploid. Your secondary spermatocyte is also haploid. So this is the process of your spermatogenesis. For this we need increased production of LH as well as FSH, which are your gonadotropins that are released from your anterior pituitary, that is your adenohypophysis. Okay. Hi Sureka, hello. So thank you so much for joining my lecture. So please do like, share and subscribe. Fine. So then we went on to talk about the structure of the sperm. I hope you remember the structure of the sperm. The sperm has a head, it has a neck, it has a middle piece and a tail. The entire structure of the sperm is surrounded by your plasma membrane. The head portion is supported with your acrosome that supplies enzymes for the process of fertilization. Then you have the middle piece, which happens to contain the mitochondria, which supplies the sperm with the ATP, and the tail enhances with the motility of the sperms. So this is about your spermatogenesis. Then we were talking about your oogenesis. So unlike your spermatogenesis, oogenesis begins at the embryonal stage itself. So each of the fetal ovary, has almost 1 to 2 million gamete mother cells. So these are known as oogonia. So once again, oogonia is plural, oogonium is singular. Okay. So these oogonia, it's a fixed number. So it does not increase after birth. So how much ever is formed at the time of embryonic development, that is it. So now this oogonia, at the time of birth, it tries to undergo meiotic division. But unfortunately, it gets arrested at the prophase 1 of meiosis. So the resulting product that we get is your primary oocyte. Okay. So this primary oocyte is surrounded by a layer of granulosa cells. So these granulosa cells are known as primary follicles. Done? So we have the primary oocyte, which is surrounded by the granulosa cells, which is your primary follicles. So a lot of primary oocytes surrounded by the primary follicles degenerate. So at the time of puberty, so this is at the time of puberty, so nearly only 6,000 to 8,000 remains, right? So now this further completes the first meiotic division. No, it does not complete. So now what happens? So further it undergoes, so at puberty, so this is the amount that is remaining, okay. So now your primary oocyte further is surrounded by a new layer of cells, that is your secondary follicle. Your secondary follicle has a new thicker layer. So this is differentiated into the outer layer and the inner layer. So slowly your primary oocyte is surrounded by the tertiary follicle. This has a fluid-filled cavity that is known as antrum. Okay. So now at the time of puberty, so here is where puberty comes. So this completes its first meiotic division. So it forms your secondary oocyte. So here your secondary oocyte is surrounded still by tertiary follicle. So gradually your secondary oocyte, so it also forms a polar body. So this polar body degenerates. So your secondary oocyte now forms a layer around itself. So this is known as zona pellucida. It also has another layer of cells like this. So these cells are known as corona radiata. Okay, so it is still surrounded by now it is surrounded by what is known as Graafian follicles. Okay, so now your Graafian follicles rupture and it releases your secondary oocyte. Here? Yes? Yes, corona radiata. Okay, I have written 6,000 to 8,000. Yes, it is 60,000 to 8,000. So you have almost 1 million at the time of birth, but here it becomes 60,000 to 80,000. Yes, absolutely right. So here you have to know the ploidy. So oogonia is 2n. So primary oocyte is also 2n because it has not completed the division. So it is still 2n. So here it becomes haploid. This is also haploid. So this is haploid. This is also haploid. So this is your oogenesis process. Now we will talk about your menstrual cycle. So menstrual cycle occurs in your human female, right? So it is a 28-day cycle. So it starts from day 1 or day 0 to day 28. So it has 4 phases. So we know on the midday, that is the 14th day, something important happens, that is ovulation. Right? So the start of your menstrual cycle is known as menarche, and the end of your menstrual cycle after an age of about 50 years is known as menopause. So this happens each month. Okay. So what are the 4 stages in your menstrual cycle? First is your menstrual phase. Then you have the follicular phase, or it is also known as the proliferative phase. Then you have the ovulatory phase or the ovulation process happening. The last one is your luteal phase or your secretory phase. So your menstrual phase happens from the 1st day to the 5th or the 7th day. So your follicular phase happens from the 7th day to the 14th day. So exactly on the 14th day, you have your ovulatory phase. So this is from the 15th to the 28th day. So what happens here? So in your menstrual phase, your endometrium that is responsible for implantation, so that disrupts and it comes out through the external male genitalia and vagina has bled. Okay? Yes? Yes, Shimpi, please ask. So follicular or the proliferative phase. So here in the follicular phase, so anything that disrupts has a magical property in your body to repair itself. Right? So here your endometrium disrupts, here it repairs itself. At the same time, the Graafian follicles develop, the follicles develop to get ready to release a ovum. So here the secondary oocyte is released. So in your follicular phase, you have increased concentration of LH and increased concentration of FSH. So at one point, the concentration of LH is so maximum that it is called as the LH surge. So this causes the rupture of the Graafian follicles. So the Graafian follicles rupture, so releasing the secondary oocyte. So this releases the secondary oocyte. Okay, done? Yes, Dharini, good evening. So this is what happens. Now, once it releases, it awaits fertilization. If there is no fertilization, it reaches the uterus and along with the endometrium, it tries to come out during the menstrual phase. So here you have your endometrium getting prepared for fertilization. Here you also have the corpus luteum secreting progesterone. So progesterone is a pregnancy hormone that is secreted by corpus luteum. So corpus luteum is the one that is developed after the remnants of the Graafian follicles, right? So this was your menstrual phase. Now we are going to talk about fertilization and implantation. Now that the gametes are ready, the male and female gamete, and how it is ready, how it is designed to produce a gamete, so all the story we have seen. Now, how does the actual process of fertilization take place? So let me tell you, though we have two gametes, the gametes have to meet, right? So only then fusion can occur. So how are they going to meet? So point number one, the male gamete has to reach the female passage, right? Because here we know the sperms are motile, the eggs are not motile enough, right? So the male sperms have to reach the female passage. So at the time of sexual intercourse or what is known as copulation, the male ejaculates the sperm into the female passage, right? So it is inserted into the vagina and it's ejaculated there. So from there the sperms move into the ampullary region. So ampulla is the region in your oviduct of your female reproductive system. So ampulla is the space where fertilization is going to happen. One more thing, your sperm and your ovum. So how many polar body? Is it 3? As of now, we have seen only one polar body. So at the time of puberty, your primary oocyte completes meiotic division 1 to see what your secondary oocyte and a polar body. So as of now, we have seen only one polar body. Okay. So now it is that the law of universe that the sperm and the egg has to reach the ampulla at the same time. So it has to be simultaneous. Just like in your sexual reproduction in flowering plants, we saw that the female flower, the stigma has to become receptive, the pollen has to get matured at the same time. There should be a sync, right? So here also, your egg from the female ovary and the sperms has to reach the ampullary region of the fallopian tube at the same time. Okay, so that is why not all copulations, not all sexual intercourse leads to fertilization, because that sync should be there between the sperm and the release of the egg. Okay, yes, a very happy good evening, Swami. So welcome to my lecture. So please do like, share and subscribe. Okay, so during copulation, the external male genitalia, that is penis, it releases the sperms into the vagina, and this process is known as insemination. So after gametogenesis, it is insemination, right? The sperms which are motile enough, they swim rapidly through the cervix region of the female and then they enter the uterus. Okay, So they quickly reach the ampullary region of the fallopian tube. So the ovary also released from the ovary also releases the ovum, which is transported by the fimbriae of the infundibulum into the ampullary region of the fallopian tube. So ampulla of the fallopian tube is where fertilization takes place. Fertilization occurs only if the sperms and the ovum are transported simultaneously. So this is the reason why not all copulations need to lead to fertilization. Lead to, not need to. Okay, so the process of fusion of the sperm with the ovum is known as fertilization. So I hope you remember, so we have your fallopian tube. So this has finger-like structures. So this is your ovary. So here we have your uterus. This is attached to the uterus with the help of ligaments. Right? So it is like this. We have another here. Right? So this is our uterus. So this is your ovary. So this entire thing is your fallopian tube. It is divided into many parts. So this portion is your infundibulum, the portion near to the ovary. So infundibulum has fimbriae. So this helps in collecting the ovum. Okay. So here this is your ampullary region. So this is isthmus. So this is where fertilization occurs. Okay. So how does it occur? So that is the next question. So first we have your sperms. So sperms reach the ampulla. Suppose assume the ampulla also has an ovum waiting there. Okay. So now how is the structure of our ovum? So we have the secondary oocyte. The secondary oocyte is surrounded by a space known as zona pellucidum. It is surrounded by a group of cells like this. So this is known as corona radiata. Right? So this is zona pellucidum. So this is corona radiata. Suppose both of them are in the ampullary region. So now a sperm comes here. Suppose this is the sperm. When the sperm comes and interacts with the zona pellucidum, so your sperm interacts with the zona pellucidum, then it causes a change in membrane permeability. Okay, with the help of the enzymes present in its acrosome. So this ensures that your zona pellucidum does not allow any other sperm to enter. So this ensures only one sperm can fertilize one ovum. So this is what this ensures. Now, the sperm reaches the cytoplasm of the secondary oocyte. Okay. So, now what happens next? So, if you can see, it is like this, right? So, the secondary oocyte, this is your secondary oocyte. So, this is your zona pellucidum. So, this space is your perivitelline space. The space between your secondary oocyte and your zona pellucidum. So your sperm comes in, interacts with the zona pellucidum, changes its membrane permeability and ensures only one sperm can fertilize one ovum. Okay. The moment the sperm enters the cytoplasm of the secondary oocyte, so here the sperm is now. Right? Yes, of course, here we have the perivitelline space. Here we have the zona pellucidum. So here is our perivitelline space. So this is our zona pellucidum. So this is also another interesting function of zona pellucidum which I will tell you. So please hold on. So now here are sperm is. The moment sperm reaches the cytoplasm of the secondary oocyte, now the secondary oocyte completes its second meiotic division. So this completes second meiotic division. thus forming a oocyte plus the second polar body. So first polar body was formed when the first meiotic division was completed. Now it is the second polar body. So this oocyte is haploid. So of course your secondary oocyte is also haploid. Now your oocyte fuses with the sperm. So when it fuses, so this is haploid, this is haploid. So they fuse together to form your diploid zygotes. Right? It is still surrounded by your zona pellucidia. So zona pellucidia's function is that till the time the zygote reaches the uterus, it does not support implantation. So this passage it has to travel from the ampullary region till the uterus. So anywhere in between it should not get implanted, right? So zona pellucidia prevents implantation anywhere between the ampullary region and the uterus. Okay, so that is the function of zona pellucidia. So this is the process of fertilization. So the oocyte completes its second meiotic division just prior to the fertilization by the sperm. It completes its first meiotic division at the time of puberty. Clear everyone? Yes? Okay. Now, how does implantation happen? So let me tell you, the zygote is still in your ampullary region. Ampullary isthmus region is still in the ampulla of the fallopian tube. Okay, so it takes ectopic pregnancy is due to zona pellucidia. So ectopic pregnancy is, yes, when your zona pellucidia is not healthy enough, it can lead to miscarriage. That is, implantation happens in the fallopian tube. Okay, Antonia B? Yes. So your zygote, as soon as fertilization is done, it is still present in the ampullary region of your fallopian tube. So the zygote to reach the uterus, it takes 5 to 7 days. So what happens during the 5 to 7 days? So point number 1, your zygote undergoes series of divisions. So this series of divisions is called as blastomeres. So your zygote undergoes 2-cell division, 4-cell, 8-cell, 16-cell division. So when it reaches the 16-cell division stage, it is known as your morula. So 16-cell stage of blastomere is known as morula. Okay. Hi Satyendra, a very happy good evening. So thank you so much for joining my lecture. So please do like, share and subscribe. I hope your preparation is going on really fine. So thank you so much for your comment on my Saturday's video. So it was just below the video. So I was seeing this morning. So I wanted to listen to my own talk this morning. So I was opening the 5 ways to improve presence of mind, and just beneath the lecture I found Satyendra's comment. So thank you so much. So this gives me a lot of motivation to do a lot of motivational videos with you. So Anthony B here also hinted that he also wants a motivation video every Saturday. So done, no issues, because I enjoyed doing motivational sessions than teaching biology. So I will make sure at least once in 2 weeks I will bring a motivational video. Okay. Thank you so much guys. So this is the process of implantation. So your zygote undergoes a series of division. So this series of division is blastomeres. So basically it undergoes 2-cell division, 4, 8 and 16. So the 8 to 16 cell stage is morula. So by the time it is in the morula stage, it has reached the isthmus junction. After this, it forms what is known as blastocyst. So it is a cavity-like. So what is in this cavity-like? So if this is your zygote, it forms an outer layer of cells and it also forms an inner mass of cells. So now your zygote is something like this. Okay. So this is your trophoblast. So this is blastocyst stage of your blastomere. Now the zygote is differentiable into trophoblast and the inner cell mass. Okay. So now your inner cell mass differentiates into 3 embryonic layers. So one is ectoderm, the other one, the inner one is endoderm, and the middle one is mesoderm. Right? When it differentiates into these 3 layers, it is known as gastrula stage. By the time these 3 germ layers form, your tropoblast gets implanted on the endometrial layer. So tropoblast gets implanted on the uterine wall, that is on the endometrial layer. So now you understand when we call as blastocyst, when it is differentiable as tropoblast and inner cell mass, this state is known as your blastocyst. Okay. In situ and ex situ conservation. Yes. So I will explain it to you at the end of this lecture. Okay. Yes. So Anthony, are we clear? So when it is differentiable as tropoblast and inner cell mass, it is called as blastocyst stage. Okay, so when it enters the blastocyst stage, your tropoblast is ready to get implanted on the uterus. At the same time, your inner cell mass differentiates into these 3 layers, and hence it is known as gastrula. Okay, so your tropoblast helps in the implantation of the zygote onto the uterine wall, and a layer of maternal tissue, that is the uterine tissue, surrounds the tropoblast. So implantation, so the 8 to 16 cell stage is morula. So it undergoes a series of division. All the series of division is your blastomere. So especially the 8 to 16 cell stage is your morula, right? So morula then enters into the blastocyst stage where it forms an outer tropoblast which attaches to the endometrium of the uterus. The inner cell mass that differentiates into the embryo, it basically differentiates into the 3 layers. So it is known as gastrula stage. So now implantation has happened. So pregnancy will occur. So this tropoblast layer, this is tropoblast and this is your inner cell mass, right? So tropoblast gets implanted on the endometrial layer of the uterus, and immediately a maternal tissue or the uterine tissue surrounds them. So this is your uterine tissue which surrounds the embryo. So quickly a tropoblast forms chro-ionic villi-like structures. They are finger-like projections. It forms structures like this. Exactly, if this is your uterine wall, it forms structures like this on the uterine wall, which later gets converted into placenta. So what does placenta do? So placenta separates the mother from the fetus. So if this is placenta, so this is fetus and this is your mother, the maternal blood. So after implantation, we no longer say it as zygote, we call it as embryo. Okay. So now what connects the placenta to the fetus is your umbilical cord. Fine. So what is your amnion? What is your amniotic fluid? What is your amniotic cavity? So we will store that for the next lecture. Okay. So now we will stop here. So this is how implantation happens, if you can see. So the secondary oocyte is released. So the zygote is formed. 2-cell stage, 4-cell stage. The 8 to 16 cell stage is your morula. So this entire division is known as your blastomere. So after your morula stage, it forms your blastocyst. So your blastocyst is when you have a tropoblast and an inner cell mass. So it quickly gets implanted here, right? So it forms a finger-like structure, that is your chro-ionic villi, which develops into the placenta. So this is how implantation happens. So pregnancy and embryonic development, I will continue in the next class. So your Swami wants me to explain what is in situ and ex situ conservation. So in situ means on site. So on site conservation is in situ. So when you conserve a particular organism on its native site, that is in situ. So in situ includes your zoological parks. Sorry, it includes your national parks. National parks, the first park being your Jim Corbett Park. So your biosphere reserves, your sanctuaries, so then you have your hotspots. Everything you do it on the same place. So your sacred groves, so all this is in situ. Okay, Swami. The next one is ex situ conservation. So ex situ is off-site. So ex situ conservation is off-site conservation. So like your tissue culture technique, your zoological parks. So for the zoos, where do you bring animals from? From the forest, right? Yes. So then you have your seed banks, etc. So this is the difference between in situ and ex situ conservation. Okay, Swami, is this clear? And through baby, yes, so it is IgG that gets circulated during your placenta, or through your placenta the antibody that is circulating is IgG. So IgA is through your colostrum. Okay, yes, so cryopreservation also comes here. Okay, Swami, done? Yes, clear everyone? So we will stop right here and we'll continue with the topic. Maybe on Wednesday I will try to finish off. So tomorrow we'll try to finish off your chemical coordination and integration. So I don't want to extend because you guys would have a timetable. So 6 o'clock you have to start the preparation of something. So because I am late, I don't want to extend your time. So please carry on with your timetable. So we will complete the class, no issue. So I will revise all the chapters. Yes, I know you have only 15 days and plus 10 more days, but still we will complete on time. Okay. So fine, thank you. If you genuinely like my way of teaching, so please do like, share and subscribe. So don't forget to be there tomorrow at 5 p.m. because we are going to say a goodbye to human physiology. So very soon we'll have a mentee in human physiology. Okay. So thank you so much for your cooperation. So this is me wishing you more and most. So this is me wishing you abundance of happiness. So thank you so much and have a great evening. Bye and take care.